Hello, everyone. I'm really excited to share with you today some research we've been working on for a long time that finally came out late last year. This is, of course, talking about dissolved organic matter, permafrost zone. I want to recognize, first of all, the giant list of co-authors. This was only possible thanks to a large international collaboration. And also to our funding sources. This was supported by the National Science Foundation through the LTER program, as well as many funding organizations around the world. So each of our teams was supported differently. Now, this, as I mentioned, this research has come out, was published in Global Biogeochemical Cycles with Ethan Rologos, the lead author. So take a look at that. I'm not going to be able to get into all of the details. But why should we care about dissolved organic matter in permafrost ecosystems? Well, as you're probably aware, this area contains a huge amount of organic carbon. This is just a compilation we put together of some of the different pools of carbon. Altogether, they store around 2.7 trillion tons, trillion with a T, of organic carbon. Globally significant pool, larger than all these other things, even recent estimates of the amount of organic carbon in marine sediment. So as climate change progresses, this is a nice figure by Schaefer and others, causes thickening of the active layer through time that exposes organic matter to decomposition and can produce carbon dioxide, methane, we also know nitrous oxide, which then accelerates the warming. Now, this is a nice figure, but it, of course, leaves out several dynamics. The first one is nutrients. It's not just carbon that's stored in these permafrost ecosystems. There are huge stocks of nitrogen, 50 to 100 gigatons, still a big range. We don't have as many estimates of nitrogen. That's in both organic and inorganic forms. What's going to happen to that as it gets released? And even less is known about the phosphorus that's stored there. So the other thing that's left out of this kind of conceptualization is what can happen laterally. So we absolutely have the vertical fluxes of gases. This is just a big thermokarst feature in Siberia. But things are moved laterally as permafrost degrades, both nutrients and organic matter, much of which is biodegradable. Not all of it. Some areas it's not very yummy carbon that's released, but in many areas, particularly with old permafrost, like Yedema, there are high concentrations of organic carbon that is extremely bioreactive that can be released. So we want to know what effects is that happening, both on local ecosystems and the global permafrost climate feedback. Now let's imagine that this river, this is the Sagamaniirtok River in northern Alaska. Let's imagine that it's the entire permafrost circumpolar watershed. There's around 100 megatons of carbon that's transported from land to water. These are inferred numbers. That's why I've got the 2 question marks there. We need a lot more research trying to understand the controls on the lateral transport of that dissolved organic matter. But if we look at what we can observe at the rivers and then estimate how much is likely removed, we end up with this figure of about 100 megatons of carbon a year. That's a large flux of dissolved organic matter, more than 10% global dissolved organic matter flux. And then we have better estimates of how much comes out, 36 megatons of carbon a year. Now, the amount of permafrost dissolved organic matter that's going to be released is smaller. We're talking about 3 to 20 megatons carbon per year. These again are inferred, lots of question marks there, but this is how much may come out by the end of the century, especially from those large thermokarst features. In any case, it's smaller than the modern DOM moving through the system. So our fundamental question in this study was, how might this smaller pool influence the larger pool that's traveling through surface waters? How will bioavailable nutrients and organic matter affect the turnover of this modern background DOM in permafrost water networks? We approached this, we had 2 kind of mechanisms that we zoomed in on that we were thinking about. The first one of how these 2 organic matter pools could interact. To understand the first one, let's take a step back and say, DOM is the source of most nutrients, both in terrestrial ecosystems and aquatic and marine ecosystems. Remember, the dissolved organic matter gets broken down and mineralized. That releases inorganic nitrogen, phosphorus, and other nutrients that are essential for photosynthesis. So when you have those nutrients there, that can stimulate photosynthesis and production of fresh DOM in these different environments. However, the nutrients also can feed back and influence the way that the DOM is being broken down, how quickly, what kinds of DOM are being broken down. These are nutrient effects. They either can stimulate and accelerate the rate of breakdown or slow it down. Likewise, this fresh DOM, which is bioavailable, can have priming effects on the background DOM. This the most famous effect is accelerating the DOM breakdown, but it also can slow down in some situations. Lots of interesting enzymatic and thermodynamic reasons for this. I like to think of it in psychological terms. You know, the microbes are using the DOM for both energy and nutrients, depending on what they need. Availability of energy or nutrients could make them not interested in it or make them more able to break it down. So again, our question is, as you have permafrost that's degrading, it's releasing both nutrients and bioavailable DOM. How is that going to affect the background DOM? We approached this problem with an international collaboration. We found 7 teams of researchers from around the permafrost zone. They sampled 3 sites each, 3 to 4 sites each actually. So there were 23 sites total, nested in 7 regions. Here's just a map showing these major watersheds and then also the location of these sites. Notice that we included the Tibetan Plateau. So we had collaborators in China, Yuan He Yang and his team that worked on that. We collected these samples in the late summer, early fall. That's the time period where permafrost degradation may be highest. So the relative amount of permafrost material in these waterways would be highest. We followed a common 28-day dark incubation based on kind of its tried and true methods pioneered by Jorin Vank and others. We had 4 treatments. The background chemistry, we added acetate. Actually, we had 3 levels of added acetate, 3 levels of added nutrients, and then a combination of all of those together to see how they affected the background DOM breakdown. Now, we picked acetate as our biolabile carbon source, both because we could quantify it independently using ion chromatography, but also because acetate's actually one of the most common things that's released from these ancient permafrost deposits. It's produced during the anoxic or anaerobic metabolism in those really cold conditions. Fascinating stuff. Look at the microbiology of permafrost to learn more about it. Because we were really excited to get these samples from all over the world, we did a suite of analyses. We quantified, of course, the DOC concentration, background nutrients, and a lot of other chemistry. We also used EAMS analysis to look at the DOM optical properties, absorbance, and fluorescence. And then we used FTICR. Please don't ask me what it stands for. Lots of Fourier transform and fancy words. But a molecular analysis that actually could break it down and look at some of the components, the composition of that DOM itself. So let's jump right into the results. We observed variable DOM biodegradability, but consistent negative priming. Now let me orient you here. We've got on the top panel our day 7 results, the bottom panel our day 28 results, and then we're expressing this in change of DOC over that time frame. So if it's near 0, there wasn't much change, and then this is a proportional rate. So these the sites that lost the most, this is in northern Siberia, they lost up to 40%. Now, these vertical lines separate out the different watersheds, and then we've organized it in each case from smallest to largest. So I'm just going to focus mainly down here. This means that as you're moving longitudinally down the watershed, no change here in interior Alaska. It increases at this site, can't remember what NC is, sorry, Northern Canada just came back to me. Sorry, Suzanne. And basically what I want to emphasize here, there's a variety of longitudinal patterns. So it's not at all the simple story of, yeah, as you move from the headwaters down to the main stem, you have a decrease in DOM biodegradability. we see a variety of different patterns that can happen. But then when we look at the difference between the control treatment and these other treatments, we observed relatively consistent negative priming across the sites. So if there's a negative, if there's an asterisk here, that means that for day 28, we saw less breakdown of the material than we did in the control treatment. So pretty interesting result there, not what we expected. Kind of good news at some level if you're thinking about the processing and release of greenhouse gases from Arctic waterways. However, remember, this still means that there's a change in the availability of the material. So it could have different local effects in aquatic and estuarine ecosystems, food webs. Now, there were larger negative effects for sites with more biodegradable DOM. I'm just going to focus down here on the day 28 results, but basically, if we had a greater proportion of DOC that disappeared, so this negative proportion here, then we were more likely to see the negative priming and negative nutrient effects. This is really interesting. It maybe means that when you've got plenty of biolabile stuff around, the microbes aren't going to the trouble of breaking down the background DOM. Particularly, we saw stronger correlations for the nutrients and combined acetate and nutrients. Surprisingly, we saw consistent molecular structure across these different sites. Tibetan Plateau, Northern Alaska, Boreal, Scandinavia, Yedema, Siberia, they all look the same molecularly, with really strong presence of unsaturated phenolics with low OC ratios. That represented 60% or more of the DOM, many of these sites. And then they've all followed the same pattern. So this could either be kind of the concept of a universal DOM structure that has been proposed by some groups, or it could be a permafrost zone specific filtering where at low temperature you have certain compounds that are preferentially removed or produced, and so you get this common signature. I won't get into that. We saw more diversity in the optical properties, and then unexpectedly we saw colored DOM loss when you had added nutrients. This is really important because CDOM is the sunscreen of aquatic ecosystems. It regulates photosynthesis and a bunch of other things. We also observed nutrient limitation of acetate breakdown. So the acetate, even though it's super yummy and biodegradable, it didn't get consumed except for when there was adequate nutrients, either the background nutrients or the added nutrients that were there. So in summary, there was variable BDOM. We didn't see strong longitudinal patterns. There were consistent negative nutrient and priming effects. The nutrient addition triggered breakdown of CDOM, and that has been observed by another paper that came out this year. If this is a general pattern, that's really important for primary productivity and also microorganisms and other things going on in aquatic ecosystems. The labile DOM uptake was nutrient limited based on the lack of uptake of acetate. All of this underlines how to understand the carbon balance of the permafrost zone. We really need to get a better grip on what's going on with nutrients, especially the lateral release of nutrients. We need to understand that in soils, in the waterways, and all the way out into the marine environment. Thank you for your attention.